Can you see?
Yes, ma'am.
Okay. We have learned the transport. We were talking about transport
Yes, ma'am.
in the plasma membrane.
Yes, ma'am.
The first one is diffusion.
Yes, ma'am.
In the diffusion, what will happen?
The particles will move from a place of higher concentration to its lower concentration until the concentration is equal.
Yes. Well, until the equilibrium is maintained.
Yes, ma'am.
Then in osmosis, what is osmosis?
Osmosis is the movement from water from a place of its higher concentration to its lower concentration across a semi-permeable membrane until equilibrium is reached.
So you see, there are two types of osmosis.
Yes, ma'am.
One is endoosmosis, the other one is exoosmosis. In endoosmosis, what will happen? The water will move in. Okay.
Yes.
So it is called endoosmosis. And if the water moves out, it is called exoosmosis. Osmosis. Okay. And there are three types of solution. Okay.
Yes.
Isotonic solution. Here in this case, I have taken. There are two cells. Okay, animal cells and plant cells. Both the cells are put in isotonic solution. When animal cell is put there, what will happen?
The animal cell, it will shrink. It will remain the same.
Yeah, isotonic solution, always the concentration outside, okay, in the environment and inside, it is the concentration of water is maintained.
Yes, ma'am.
The amount of water getting in is equal to amount of water going out. Okay, so
yes.
It will be maintained and there will be no net movement of water.
Yes, ma'am.
There will be movement, but there won't be net movement. Okay.
Yes, ma'am.
And here in the hypotonic solution,
is where the water concentration outside is more than inside of the cell. So the water will enter the cell and in plants, the cell won't burst because of the cell wall. Whereas in animals, it will burst.
Animal cell will burst because there is no cell wall. And in this type of solution, the concentration is more. Okay.
Yes.
Solute concentration. Okay. And so here this is blood cell. So that process, the breaking of that cell is called hemolysis. Lysis means breaking. And in the hypotonic solution, the concentration of water is more, okay, inside the cell.
Yes, ma'am.
And outside, there is less. So water will come out.
Yes, ma'am.
And this is called prenation or shrivel.
Yes.
Shrivel. And in this case, which osmosis is occurring here? Which type of
exosmosis. In
endo, hypo, endo, okay?
Okay, ma'am. I thought you were about hypertonic.
Yeah. Endoosmosis, okay? And
yes.
Here,
exo.
Okay?
Yes, ma'am.
Now, let's talk about the plant cell. In the plant cell, in the isotonic solution, the cell will be normal, okay?
Yes, ma'am.
And in hypotonic solution, what kind of cell will be there?
The cell will swell up, but it won't burst because they exert same amount of pressure on the cell wall. The cell wall exerts the same amount of pressure on it and it maintains the...
That's why cell will be turgid, okay?
Yes, ma'am.
And in the case of this hypertonic solution, water will move out. So the protoplasm, the living part of the cell, will move away from the cell wall. That is called plasmolysis.
Yes, ma'am.
So this all are the movements, okay? And now you have seen the structure of plasma membrane, right?
Yes, ma'am.
This small non-polar particles, right? It will move quickly, okay? Their transport will be quick.
Yes,
small non-polar molecule like oxygen, carbon dioxide, okay? Diffusion.
Ma'am, what are polar particles?
Non-polar.
Yes, ma'am. What exactly are they?
That is 1 minute. I'll tell you.
Yes.
There are two types of molecules. Okay, polar and non-polar. Non-polar, there won't be any charged particles.
Okay.
And in polar, there will be charged particles. Okay.
Yes.
So here, small non-polar particles, right? They will be able to move very fast through the lipid bilayer, okay, to the plasma membrane. It is easier to transport. And small polar molecules like water, it will be also moving, but it will be slow, okay.
Yes.
And large and non-polar molecules like vitamins, they will be also moving, but it will be very slow.
Yes, ma'am.
This large and polar and highly polar and charged ions. Okay, these things, right, they require transport protein to transport them. Okay,
Yes, ma'am.
okay, this protein channel, okay, this will help in the transport.
Okay, ma'am.
Okay, through this only the particle will move from here or here or here, anywhere it can't move. It could move only through the protein channel.
Okay, ma'am. So those blue color structures are the protein channels.
Yes.
Yes, ma'am.
There will be different types of protein channel for particular protein. Okay.
Okay, ma'am.
And for movement, this movement, right, it involves energy. It requires energy. Okay, ATP. These movement are what? Active transport.
Yes, ma'am.
And for large molecules, there is two type of movement. Okay, endocytosis and exocytosis.
Yes, ma'am.
Okay, and here also there is requirement of ATP.
Yes, ma'am. Ma'am, so protein is a large and polar particle. Protein is highly polar and charged ion.
Hmm.
Okay, ma'am.
Now let's see. Endocytosis, we have three types. Okay, phagocytosis, pinocytosis, and receptor-mediated endocytosis.
Yes, ma'am.
Phagocytosis means eat to eat.
Yes, ma'am.
Okay, so here this happens in the white blood cell. White blood cell does phagocytosis. Okay, macrophages, neutrophil, these all are white blood cell, which helps to fight diseases. Okay.
Yes, ma'am.
Now you see here, this is the surface of the white blood cell. You can say macrophage. Okay.
Yes.
Okay, and this here is bacteria.
Yes, ma'am.
This surface of the what is that? Macrophage has receptors. Okay.
Yes, ma'am.
This Y shaped, you can see.
Yes, ma'am.
It binds the bacteria. Okay.
Yes, ma'am.
And this cell membrane, what will happen? It will create. It will engulf. Okay. It changes its shape.
Yes.
This is called pseudopode. Okay.
Yes, ma'am.
And the bacteria will get inside the macrophage.
Yes, ma'am.
Hmm?
Yes, ma'am.
And this is called phagosome.
Yes.
And there will be lysosome. Okay.
Yes, ma'am.
Which will have what?
Digestive enzyme.
Yes, digestive enzymes.
Yes, ma'am.
Both the thing will join and they will form phagolysosome. Hence, this enzymes will destroy that bacteria, and after that, that pseudopode will open and the remaining particle will move out.
Yes, ma'am.
So this process is called phagocytosis.
Yes, ma'am.
Here, I told the transport of large material, because this is large. That's why it will come under the endocytosis. Okay, not in the active transport, not in the passive diffusion or osmosis. Okay.
Yes, ma'am.
Next, we have pinocytosis. This meaning is meaning means to drink. Okay.
Yes, ma'am.
In the cell. Okay. This is a cell. This is the plasma membrane.
Yes, ma'am.
Okay. And here, in outside the cell, we will be having many particles. Okay. Substance and solution also. Okay.
Yes, ma'am. So if it is a solution, then won't there be like diffusion or anything else happening?
No. Here, the solution. See water, right? It requires many things. It helps many processes. Okay, it can go by itself, and it can carry the particles through the protein channels, or it can carry particles from pinocytosis also.
Okay, ma'am.
Okay, water does many things.
Yes, ma'am.
And here there are many particles here. Our cell wants them.
Yes, ma'am.
So here this thing will be created. This is called carveole. Okay.
Okay, ma'am.
This structure, this cup-shaped structure.
Yes, ma'am.
And after that, it will get inside and it will form vesicle. It will get inside.
Yes, ma'am.
And there will be protein. It will carry the vesicle inside the cell. And inside the cell, particles will come out. Okay?
Yes,
From
ma'am.
these vesicles. This is called pinocytosis.
Yes, ma'am.
Next, we have receptor-mediated endocytosis.
Yes, ma'am.
This is the cell, okay?
Yes, ma'am.
Mm, in receptor mediated endocytosis, transport of LDL, low density lipoprotein.
Okay, ma'am.
This is an example. Okay, transport of low density lipoprotein by receptor mediated endocytosis. Okay,
Yes, ma'am.
here we have receptors. Here we have receptors in the cell, and here we have what? Clathrin. It is protein. Okay,
Okay, ma'am.
and LDL will bind to the receptor.
Yes, ma'am.
And here the clathrin protein, here cup-shaped structure will be formed. Okay, together they will link up.
Yes.
This will form steady shell like this
Yes, ma'am.
tortoise.
Yes.
After that, the vesicle will be formed. Okay, this clathrin will detach.
Yes, ma'am.
And this vesicle will join with
Why is it also?
what? Endosome.
Oh.
It's a type of vesicle.
Yes, ma'am.
It allows this LDL and this receptor to split up.
Yes,
Right.
ma'am.
And receptors will again go to the surface
Yes, ma'am.
where there will be clathrin proteins. Okay, again this process will be happening, and this one LDL. Okay, here lysosome will work and it will digest.
Yes, ma'am.
Okay, understood?
Yes, ma'am.
So this all was process of endocytosis. Next, exocytosis. Endocytosis meaning the content will come inside the cell. Okay,
Yes.
whatever material will come inside the cell. In the exocytosis, the content will move out of the cell.
Yes, ma'am.
Okay, now you can see here. Here we have a nucleus, and this is endoplasmic reticulum.
Yes, ma'am.
Okay, now proteins, okay, will be carried to the Golgi apparatus through transport vesicle. Okay, from there the protein will be, what will happen here? The protein will be modified, will be packed. Okay,
Yes.
sorted. And after that the vesicle from the Golgi apparatus, okay, will come out. It will be carrying the protein.
Yes.
And it will go out of the plasma membrane. Okay, the vesicle will split, break off, and the protein will go out of the cell. Okay, this is secretory vesicle.
Yes, ma'am.
This process is called exocytosis.
Yes, ma'am.
Understood?
Yes, ma'am.
So these all were the transport across the cell membrane, how it is happening.
Yes, ma'am.
So the plasma membrane is very important. So next, we can see the function of plasma membrane. It helps maintain the shape of the cell.
Yes, ma'am.
And next, it acts as mechanical barrier and protects the internal content of the cell from leaking out. Then it provides protection against microbe and foreign substances, and it gets modified to perform different function. Okay, microvilli in intestine of human being for absorption.
Yes, ma'am.
Its semi-permeability enables the cell to maintain cellular homeostasis. That means balance of this. Any doubt?
No, ma'am.
Now let's talk about cell wall. Cell wall, it is found in plants. It is not found in animals. Okay.
Yes, ma'am.
Plants, bacteria, and fungi. In plant, these all are the organism where cell wall is formed. Okay. In plant, the cell wall is made up of cellulose,
Yes, ma'am.
pectin, and hemicellulose also. Okay. The main content is cellulose. In bacteria, it is peptidoglycan. Okay. And in fungi, chitin.
Yes.
Okay. This peptidoglycan we can also say as murin. Okay.
Yes, ma'am.
In plant cell, the outermost covering is cell wall. After that only there will be cell membrane.
Yes.
Bigger one. Yes. So this is cell wall and this is plasma membrane. Outermost covering of the animal cell is plasma membrane, and outermost covering of plant cell is
Cell wall.
cell wall. And
Yes.
this cell wall is what? It is permeable membrane. Okay, it allows all the particles to move, come in and go out.
Yes, ma'am.
And what is its function? In the hypotonic solution,
Hypotonic, it allows the cell to not burst.
and in the hypertonic solution,
In the hypertonic, the cell it shrinks out of the cell.
and what will happen?
Plasmolysis.
Okay. Now let's see. Cell wall permits the cells of plants, fungi, and bacteria to withstand hypotonic condition without bursting. In hypotonic media, the cell tends to take up water by osmosis. The cells swell up, building up pressure against the cell wall. The wall exerts equal pressure against the swollen cell.
Yes.
Cell wall help plants to transport greater changes in surrounding medium. It is absent in animal cell. This is very important.
Yes.
Next one, it has narrow pores called pits. Okay, it has pits. Through them, fine strand of cytoplasm, okay, which is called plasmodesmata, are able to cross cell wall.
Yes, ma'am.
Okay, it creates a channel, and through this, plant cell will interact with each other.
Yes, ma'am.
The cytoplasmic channel are called plasmodesmata.
Okay, ma'am.
Bridges. Next, plasmolysis. It is a phenomenon in which a living plant cell loses water through osmosis when kept in hypertonic solution. In this process, there is either shrinkage or contraction of protoplasm away from the cell wall. Okay, the cell wall, we have how many points? We have about the hypertonic solution, and then pits, connection between cells. Okay,
Yes, ma'am.
these all things you have to write.
Okay, ma'am.
Next, we'll go to the nucleus.
Yes, ma'am.
It is known as brain of the cell.
Yes, ma'am. It is called the brain because it directs all the functions and activities inside the cell.
Why does it direct? How? What does it contains?
The nucleus contains nucleoplasm.
No, I'm talking. Why are you telling that it is brain and it does all the work? It controls all the things. Why? What does it contain? What does it have that others don't have?
It has its like
DNA.
It has like many sets of things. Like when the nucleus has all of them, we call it as a true nucleus. So
I'm talking about why does it does all work? Why it is called brain of the cell? Because in the it has DNA. DNA contains instruction, information.
yes, ma'am.
Okay, DNA, it will form what? RNA. RNA will form protein. Protein will help in the structure of cells.
Yes.
Okay, and the cells, all those cells, there will be reactions, and all the organs and all the organ system will be controlled. Okay, the important is DNA. DNA has what? Gene.
Yes.
Okay, this DNA, the gene, right?
Yes, ma'am.
It is functional part of DNA.
Okay, ma'am.
This only encodes the information.
Yes, ma'am.
Now, what were you telling? What are the parts of the nucleus?
Yes.
Tell me.
Well, you first have the nuclear membrane, then you have the chromatin material, and then you have the nucleolus, and you have the nucleoplasm.
And we have nuclear pore,
Yes, ma'am.
where the transport of material between cytoplasm and nucleus happens through nuclear pore.
Yes, ma'am.
The fluid content of the nucleus is nucleoplasm.
Yes, ma'am.
And nucleolus, what does it do? It produces ribosome.
Okay, ma'am.
Ribosome produces protein.
Yes, ma'am.
So let's see. The nucleus, it is popularly known as called as brain of the cell. It is composed of double layer covering called nuclear envelope or nuclear membrane. Okay, it has numerous pore called nuclear pore. They transfer the material from inside the nucleus to the cytoplasm.
Yes, ma'am.
The nucleus contains chromosome. When they are visible as rod-shaped structure, only when cell is about to divide. When the cell is about to divide, we see chromosome. Okay.
Yes, ma'am.
In the chromosome only, we have genes.
Yes, ma'am.
It encloses a liquid substance called nucleoplasm. There will be nucleoplasm. It contains nucleolus and chromatin material.
Yes.
This chromatin material only condenses to form chromosome.
Yes.
Nucleus is a more or less round structure found inside the nucleus. It does not have covering of the membrane. It is known as factory of ribosome. Chromatin is enlarged network of long thread-like structure. It condenses to form chromosome during cell division.
Yes.
Now, chromosome contain information for the inheritance of features from parents to next generation in the form of DNA component. DNA, क्या रहता है? DNA, what does it have? DNA has genes. Okay. Nucleus also contain RNA that directs protein synthesis. In some organism like bacteria, nuclear region of the cell is poorly defined because of the absence of nuclear membrane.
Yes.
The nuclear membrane in this organism contain only nucleic acid. Such undefined nuclear region is called nucleoid. Okay.
Yes, ma'am.
Based on the nucleus, the cells are divided into two forms.
Yes, ma'am. Prokaryotic and eukaryotic.
Yes. So prokaryotic.
Yes, ma'am. Prokaryotic does not have a true nucleus, and eukaryotic organisms do have a true nucleus.
Prokaryotes have pro meaning primitive.
Yes, ma'am.
Karyotic meaning
Yes.
nucleus.
Yes.
Hmm?
Yes, ma'am. And U means true.
True. Yes, correct. And here true. Primitive meaning here poorly defined.
Yes, ma'am.
Called nucleoid. There is no nuclear membrane. And one more thing is not there in this case.
There is also no, like it does not have a chromatin material also. And there is no nucleolus.
Hmm.
And no nucleoplasm. It only has the nucleic acid.
And in the case of eukaryotes,
It has chromatin material, has nucleolus, has the nucleoplasm and nuclear membrane.
So nuclear membrane, nucleoplasm, nucleolus, and nuclear pore chromatin.
Yes, ma'am.
That's all. Let's see more difference between both the prokaryotic and eukaryotic cells.
Yes, ma'am.
Here you see prokaryotic cell. It is generally small, 1 to 10 μm micrometer. Okay.
Okay, ma'am.
And in eukaryotic cell, they are large, 5 to 100 μm. Okay. Next, in the nuclear region, the prokaryotic cell are poorly developed, no nuclear membrane, called nucleoid. This we have seen. And here, in the case of eukaryotic cell, well developed, and it is surrounded by nuclear membrane. Here, prokaryotic chromosome single. Here in eukaryotic, more than one. How much chromosome do we have?
I think 23 pairs.
23 pairs. So how many chromosome? 46.
Yes, ma'am.
Here the nucleolus absent. In the eukaryotic present. Then membrane bound cell organelles in the prokaryote cell, it is absent. And here it is present.
Yes, ma'am.
Centriole, it is present during the cell division. Okay.
Yes, ma'am.
Absent in prokaryotic cell, but it is present in animal cell.
Yes, ma'am.
Then organism where it is found in bacteria, blue-green algae. In eukaryotic, where it is found? Animals, fungi, plants, okay, all those.
Yes, ma'am.
Cell division, how it is formed? Binary fission or budding. And in case of eukaryotic cell, it forms by mitotic division or meiosis. Meiotic division, okay. Okay, now we have learned about this. In the next class, we'll learn about the function of nucleus. Then we will go to the cytoplasm. After that, cell organelles will learn, okay.
Yes, ma'am.
Okay, understood?
Yes, ma'am.
Thank you then.
Ma'am, thank you, ma'am.
Okay, bye-bye.
Thank you, ma'am. Bye, ma'am.